Kapil. Thank you. Needs to bring your presentation.
Yeah.
Good morning. So it's well established that a variety of cellular insults induce DNA damage. These insults can be therapies such as chemotherapy, ionizing radiation, UV light, or other insults like increase in intracellular reactive oxygen species or replication errors. Now, the DNA damage elicits a DNA damage response in breast cancer cells, and this could be activation of checkpoints, cell cycle checkpoints, modulation of gene transcription, or engagement of DNA repair pathways or DNA repair mechanisms, such as homologous recombination, non-homologous end joining for double-strand DNA breaks, and base excision repair, nucleotide excision repair, mismatch repair for other forms of DNA damage, including single-strand DNA breaks. And if the DNA is not damaged, cell is induced to undergo programmed cell death or apoptosis. It is also well established that breast cancer cells with defective DNA damage response and repair mechanisms are sensitive to DNA damaging chemotherapy, which has been the basis of all our treatments for a long time. Breast cancer cells with defective homologous recombination HR DNA repair mechanism due to BRCA1 mutation show greater sensitivity to poly-ADP ribose polymerase PARP inhibitor, which constitutes synthetic lethality, a lethal combination of individually two non-lethal cellular insults. ERPR HER2 amplification negative triple negative breast cancer cells or TNBC cells without BRCA1 mutation also exhibit greater sensitivity to PARP inhibitor and cisplatin. This has been previously reported. Recent studies have also shown that DNA damage response repair proteins ATR, CHEK1, BRCA1, RAD52 require chaperone association with heat shock protein 90, HSP90, to maintain their levels and activities. HSP90 keeps them inactive in native conformation. Now, I've summarized our findings in this cartoon. Well, what we showed here in our studies is that pan-histone deacetylase inhibitors induce reactive oxygen species and deplete homologous recombination proteins BRCA1, also BRCA2, and RAD52. Pan-HDAC inhibitors also deplete some of the DNA damage response proteins, ATR and CHEK1. Now, pan-HDAC inhibitors are known to inhibit histone deacetylases, HDAC6 in the cytosol and HDAC3 in the nucleus that we found, which induces hyperacetylation of heat shock protein 90, as shown in the cartoon. And this leads to disruption of its chaperone function towards BRCA1, CHEK1, RAD52, ATR, which then get polyubiquitinated and proteasomally degraded. Their levels go down. And this basically induces a BRCAness in the breast cancer cells and making them sensitive to other DNA damaging agents, cisplatin, as well as PARP inhibitors, which inhibit the alternative pathway of DNA repair, base excision repair. And so essentially combining pan-HDAC inhibitors with either cisplatin or PARP inhibitor, you're inducing a lot more DNA damage, decreasing DNA repair, abrogating cell cycle checkpoints, accumulating more DNA damage, and causing synergistic cell lethality. So summary of our findings are listed here. HDAC inhibitors induce ROS and DNA damage, but deplete the levels of DNA damage response proteins, ATR, CHEK1, BRCA1, and RAD52. And depletion of DDR proteins is at least partly due to pan-HDAC inhibitor-mediated inhibition of HDAC3, induction of nuclear HSP90 acetylation, and disruption of chaperone association of HSP90 with DDR proteins. And co-treatment with PARP inhibitor and pan-HDAC inhibitor or cisplatin synergistically induces apoptosis of triple-negative breast cancer cells. And these studies support the rationale to test the efficacy of a in vivo treatment regimen that includes a PARP inhibitor combined with a pan-HDAC inhibitor and cisplatin against triple-negative breast cancers. Thank you.